to our mailing list. So with no further ado, I'll get to introduce Professor Gaia Novarino. She's an Italian native. She received a PhD degree in developmental biology from the University of Rome, La Sapienza in Italy. During her PhD, she moved to Germany to join the Max Delbrück Center for Molecular Medicine, where she focused on the study of chloride transporters associated with human genetic disorders. In 2010, she moved to San Diego to do a postdoc at the School of Medicine of University of California, San Diego, where she studied the genetics of neurodevelopmental disorders. In 2014, she joined the Institute of Science and Technology Austria, first as an assistant professor and then as full professor. She's a FENS Kavli Network of Excellence alumni, an ERC grant holder, and a Simons Foundation investigator. And in 2016, Gaia won the prestigious Boehringer Ingelheim Friend Research Award for her research on molecular mechanisms underlying human neurodevelopmental disorders. And finally, since 2021, she's the Vice President for Science Education at ISD Austria. And Professor Novarino research interest lies in understanding of the molecular mechanisms underlying neurodevelopmental and neuropsychiatric disorders. So with this, I'll pass the word to you, Professor Novarino.
Thank you, Rita, for the introduction, and hello, everyone. So as Rita just mentioned, my lab is interested in studying neurodevelopmental disorders, and among those, some that we are very focused on are the autism spectrum disorders. And there's some, they're cut away, whatever, and some animation here probably missing because of the system we are using now, but it doesn't really matter. So autism spectrum disorders has a few of you eventually already know, are characterized by those challenges in establishing social interactions, limited communication skills, and repetitive stereotype behaviors. And so far we know that those are highly genetic disorders, in the sense that mutation in a number of different genes have been associated with the development of this group of disorders, of conditions. And so far we have over 400 genes that are really associated quite strongly with autism spectrum disorders. And so this is a very heterogeneous group of disorders, and that's why there is this say that if you have met one person with autism, you have really met just one person with autism, really indicating this heterogeneity of those patients. And because of these several groups, including ours, since a few years, what we are trying to do is try to understand what kind of bring together or how to group this is really whatever. It's a bit of an issue with with this way of sharing, but it doesn't matter. Just maybe you don't see everything. But anyway, the way that we are working on is really to try to understand how to group a subgroup of patients in a way that we can identify common molecular pathways and commonality again among them. And this is because, of course, it could ease the eventually the search for common therapies that could be effective in at least subgroups of those patients. And so over the years, for instance, what we have done is, so we and others, what we have found is that defects in synaptic plasticity, you won't see this in this slide, but the defects in this synaptic plasticity, as well as cytoskeletal organization, proteostasis, as well as transcriptional regulations, are at the basis of those conditions. However, what we know very little of is how those genetic factors that over the years we and others have identified, how do they sort of interact with a very much less understood part or ensemble of factors that are those environmental factors that could, in fact, also influence how the brain is developing. And so because of that, so we became really interested in some aspects. So overall, what my lab does is really to study how the brain develops and to look this under sort of the umbrella of autism spectrum disorders. So we are selecting genes of interest based on autism spectrum disorders, and we are looking how mutations in this gene affect the development of the brain. Because of this black box, so this environmental factor part that we still don't understand. So we recently started asking how different factors such as metabolism eventually could move or change the role of or the effect of the penetrance of genetic mutations that we find associated with autism spectrum disorders. And all of this started because a few years back, some of you that may know my sort of research path know that since some years I've been developing an interest in this gene that is called SLC75, that we first identified in a couple of families. Here are the pedigree with a number of different patients or individuals with autism spectrum disorders, motor deficits, as well as mild to severe microcephaly. So what all these patients here in red, indicating here in red, have are mutation in this gene SLC75 and all share homozygous missense mutations in this gene. And those mutations we have shown in previous work that are indeed mutations, not just genetic variants in the sense that are impacting the function of the product of this gene. So what the SLC75 is doing is actually regulating the entrance into various cell types of number of different amino acids, including the branching amino acids, and those are isoleucine, leucine, and valine. And the only thing that sort of you need to know for the sake of this talk is that those amino acids are essential, meaning that they cannot be produced by any cell in our body, and so they need to be taken by through our diet, and that branching amino acids are also metabolically relevant. And so because of that, because of the condition that we identified, we realized that, and because of the function of this gene, back then we realized that we really knew very little about not only the branching amino acids, but about in general the level of the number of different metabolites in the brain. So specifically, we went back very early on when we discovered those patients and looked back at the level of the branching amino acids into the brain, and we found out that indeed we knew pretty much nothing, and for us was really quite a surprise that we had no clue about what the level is in the brain in humans, but also in a number of other mammals. We also didn't know whether the level is changing over time, which is, so is it any particular time period where they are higher, when they are more required, more utilized, etc. And in fact, this is, as I mentioned, this is also true for a number of other metabolites. So we don't know in general, we didn't know in general how those metabolites are, what is their level, and how they are changing across development. And so what we decided to do back then, so one additional piece is in fact that, looking back at the literature, we also didn't know, or we don't know, how is basically the metabolic setup of different neuronal cell types at different stages. For instance, what we knew is that very early on, neuroprogenitor cells, they are working they are relying on the production of ATP based on anaerobic glycolysis. Later on, so this is a fairly inefficient way of producing ATP. So each cycle, basically, of the conversion from glucose to lactate produce about 2 molecules of ATP. So this is a very ineffective or inefficient way of producing energy. So as soon as later on the neurons start firing when they really require much more energy, they switch to a different metabolic modality whereby they are using oxidative phosphorylation. That is a way more efficient way for producing energy. So each cycle, in this case, produce 32 molecules of ATP. But in this case, neurons are requiring the support from the glial cells that are providing to the neurons not only glucose, but also pyruvate and lactate. So what instead happens in sort of in between those different states of the neurons. So when the neurons eventually they start to be active, but there is not yet the support from glial cells simply because they did not develop, they are not developing the same exactly at the same stage. It wasn't clear. So what we decided to do back then it was to perform a metabolic analysis of the cerebral cortex from mouse at different developmental stages. So starting from E14.5, then postnatal day P2, and then in adult animals P40. And what we immediately noticed is that, and you can see this in this principal component analysis here on the bottom, is that just using the levels of the metabolites, so we quantify approximately 500 metabolites, by just using the level of those metabolites, we were able to, in an unbiased way, to separate the samples obtained at the different developmental stages from the cortex, indicating that what is usually referred as sort of the metabolic state of the brain eventually is really changing quite dramatically quite dramatically across development. So you can really separate those samples just based on the level of those metabolites. What you can also see on the right side, so this heat map indicates, so each row is one sample, with the first 4 being samples obtained from E14.5, the next 4 P2, and the last 4 P40. What you maybe can realize is that in within one time point, each animal, so the level of the metabolites within each time point is quite conserved also across animals. And this was, in a way, I think it was a little bit of a surprise, because despite we were, of course, quite careful in obtaining the samples, for instance, at the same time of the day, and having the proper controls, it was quite a surprise to see that, in fact, there is a really very high degree of conservation across animals. So again, indicating that those are not just metabolites that are changing sort of randomly, but is a quite a well-conserved program. And so next, to really ask what this a conserved program is, and so to sort of assign not just a quantitative but also a qualitative meaning to the changes that we were seeing, we did a step further and cluster the different metabolites that we quantified in groups based on the trajectory that they follow across time. So namely, what we classify, what we found are 6 different clusters, grouping metabolites that follow different trajectories over time, so at the time points that we measure, so E14.5, P2, and P40. And so, for instance, what you see is in the cluster 0, we classified metabolites that are relatively higher at the E14.5, and then they have a drop postnatally at P2, and then they eventually kind of go back at the adult stages. The next cluster, in cluster 1, we have instead metabolites that are overall increasing over time across development, or for instance, in cluster 3, you have an example of a group of metabolites that are rather decreasing over time throughout development. And so in order then to understand whether those clusters were enriched to specific metabolites, we then perform a GO-term analysis. And so what we found is that, for instance, just to give you a few examples, the cluster 1 was enriched, so that cluster 1 is that cluster that has metabolites that are increasing in level over time. it was enriched for metabolites that were related to CAMP signaling and as well as neuroactive ligands. And this makes a lot of sense because practically, so among those you will find also GABA, glutamate, and so on and so forth. So, for instance, on neurotransmitters practically. And so this makes a lot of sense because, of course, as I mentioned also before, and as you probably all know, the brain is maturing. So at the beginning, the neuroprogenitor cells, they are not really active. And so as the brain matures, more neurons are forming. And so more of those neurotransmitters or neuroactive metabolites are produced. So this was sort of acting as our positive control. On the other side, one other enrichment that we found, and this was very unbiased, and so for us was surprising but nice to find, is that cluster 3 that has those metabolites that are decreasing over time was enriched for valine, leucine, and isoleucine related metabolites, and those are the metabolites that eventually are also transported by SLC75. That, if you recall, this was the gene that we identified back then as mutated in patients with this form of autism spectrum disorder, microcephaly. And so here I would like just to point out that although it is nice to classify and look that those metabolites are changing trajectory over time, we could not really assign a meaning to those trajectory in terms of functional meaning to those trajectory yet, because in contrary to, for instance, transcriptomic analysis, where you know that an increase eventually in mRNA level of a certain gene or a certain transcript is eventually read as a function of that gene at that time point. In the metabolomic analysis, a bit more complex because a reduction of a metabolite could mean either a less uptake or less requirement for that metabolite, or also a more need, so a higher use of that metabolites, and so that's why it's dropping. So in order to really understand a little bit more what this meant, we had to go back and to do some functional work. And so our hypothesis there back then was that, in fact, at this point, branch and amino acid, they are dropping at P2 because maybe neurons that lack this glia support is utilizing branch amino acids as a way to produce energy. So at this point, when neurons start to be sort of active, lacking the glia support, so the pyruvate and lactase support, uses branching amino acids as a way to produce energy. So we therefore ask what happens if those branching amino acids are lacking, or what happens when you're changing the branching amino acid levels in the neurons, and how this affect the metabolic program of the neurons, and how does this affect the brain development. So to do practically a bit more functional work in understanding how and what does it mean, this decrease in this level of branching amino acids over time that we observe by a simple metabolic analysis. And so in order to understand that, we went back to our initial discovery, namely that SLC75 mutations leads to a specific condition. And so because we have SLC75 condition on animals in my lab, what we did is simply deleting SLC75, this time specifically from the precursor, from neuron precursors, and so all the excitatory neuron cell lineage of the forebrain. And so with these mice in the ends, we went back then and really tried to understand how limiting the access, or the entry of the branching amino acids inside neurons affects eventually the metabolic program of those cells. And so we went back and performed again the metabolic analysis that we performed initially in control animals to try to understand how the metabolic state of the brain changes in those mutants. And so here you have now on the left side, you have the same clustering that I have shown to you before, simply with a different type of graphic. This is itinerary plot where each dot that you see inside the triangle is one metabolite, and the position of that dot is determined by the ratio of the level of that specific metabolite at the 3 different time points that we measured. So in practice, you see that we color the different metabolites, so we assign a different color to different metabolites based on the more or less on the position in within this triangle. So indicating that each of these metabolites with different colors have a different trajectory, and those correspond to the clusters that I have shown to you before. And on the right side, instead, what you see is the same representation, now representing the metabolites that we detected also in the mutant animals that we generated, but depicting only the metabolites that are changing trajectories in over time in the knockouts or in the mutant animals. So practically what you can see is now the one metabolite, so where I'm pointing, I don't know if you can see my cursor, in fact, you can't, right? So the position of, for instance, this, so the knockout here is always this turquoise type of color. So each metabolite, and the gray color instead is the position, meaning the level of the same metabolite connected to the line, to the turquoise one, in the wild type. And so practically what we are showing here are just those metabolites, so not all the metabolites that we quantify, but just those metabolites that we quantify in the knockouts, and then the knockouts have a different trajectory compared to the wild type. Now, what you may be able to appreciate just from having a look at this triangle, that this is quite a number of metabolites. So they are not just the 3 metabolites that we are, or the 3 amino acids that eventually the SLC75 transporter is transporting. So indicating that despite here we are deleting a gene in the neurons for transporting the branching amino acids, in fact, what we found is that we found many more metabolites that are significantly affected by, have their trajectory significantly affected by the deletion of this gene. And so when, and I don't know if this will appear now, but when we did then an enrichment analysis to understand which metabolites are changing trajectory over time, what we found is that in the mutant, we have abnormal levels, at least at one time point, of the branching amino acids, which is somehow not surprising, of course, because here we are mutating a transporter for those same amino acids. But also on the side, what we found is that we have abnormal level of the glycerophospholipids related metabolites. And this was very surprising because obviously the SLC75 doesn't have anything to do with the lipids. And so we were really wondering why that was the case. And our explanation, so what we found is also, we did look at the data again to look really specifically at the glycerophospholipids level. And what we found is that just based on the level of glycerophospholipids, you could separate knockouts and wild type, but just at one specific time point. So namely the P2, so the postnatal stages. So practically, so what you're seeing in this principal component analysis is that at E40.5 and at P40, the different samples, so knockout and wild type samples, they are clustering together based on the level of glycerophospholipids. But at P2, they are really separated in 2 different groups, indicating that the level of the lipids is specifically different at this P2 stage. And here, this obviously you cannot see very well, and sorry about that. So what we did, we went on by checking, did a lipidomic analysis of the cell and tissues of the cerebral cortex of the wild type and mutant animals. And what we found is that indeed we have a lower amount of glycerophospholipids in the mutant brains as opposed as to the wild type. Obviously, this is very interesting because glycerophospholipids can have a number of different functions in the brain and in any cell. In fact, they have an important role in maintaining the structure of the membrane. They can have an important role as well in modifying proteins. And finally, of course, they can be used to produce energy. So the other part, and again, this is messed up somehow, but the other part that we found is that based on this analysis, we found that in the brain tissue, we had, or the mutant tissue, we had higher amounts of triglycerols. And this is extremely interesting. I mean, from this group of people actually that look at the body-brain interactions, because it's extremely interesting, because what we know is that these triglycerols cannot be produced in the brain. And so, in fact, most probably they get the liver from other tissues, for instance, the liver. And so what we thought here, and in fact, based on further proteomic analysis, we could support based on the different proteins that we see deregulated in those mutants, is that despite we are deleting the SLC75 transporter exclusively in the brain sends a signal probably of low energy, so that needs more, basically, a substrate to produce energy, and in this case, a substrate are lipids, send this signal to other parts of the body, including the liver, and the liver sort of delivers triglycerols to be used then to produce those lipids and this energy. So I think this is a really beautiful, for us, was a beautiful example, again, because we really had a control in the sense that we knew that we were deleting the SLC75 transporter just in the forebrain, and yet we will get recruiting. We were recruiting, those animals are recruiting triglycerols to the brain. So it's really, we think, a very good example about brain-to-body communication. So so our hypothesis here, it was really that at these developmental stages, so this P2 postnatal stage, when neurons are depending probably on branching amino acids to produce energy, that at this stage, if the branching amino acid level drops abnormally, like in our SLC75 mutants, the neuronal cells are starting utilizing lipids to produce ATP. And in fact, we did not find any difference in ATP because probably those lipids are really sufficient to compensate for the deficiency of the branching amino acids. Yet, of course, the question was, but what is then? What is the phenotype of those animals, and what is eventually the molecular mechanism? And so, in a way, we went back again. How does changing branching amino acid levels affect not only the metabolic program but also the brain development? So, since those lipids seems to be sufficient to compensate for the lack of the branching amino acids, at least at the energy level, what is eventually the phenotype? Do those animals still have a phenotype? And indeed, this is the case. So they do have a quite a clear phenotype, and namely, what we observe is that those animals postnatally, starting at P0, they are starting developing microcephaly. So the head size of those animals decreases or actually is growing with a different speed compared to the wild type animals. Interestingly enough, this is happening at a very, very specific time window. So it's happening in between P0 and P10. So you see practically the gray line is always the wild type, the turquoise is the mutant. And what you observe is that those two lines are kind of separate in the window between P0 and P10, but then they remain sort of parallel. So whatever happens, it happens really in within this time period. That, interestingly enough, was also the time point where we also saw this defect, specific defect in lipids, right? So we were seeing in this principal component analysis that specifically at P2, mutants and knockout and wild-type animals were different in their profile of the leaves. So, and this really caught our attention also because we knew that this P0 to P10 developmental stage may be, or actually it is a very important developmental stage or window in brain development. And namely what other people have shown before is that between postnatal day 0 and postnatal day 10 in mouse, the network is getting reshaped, and in particularly there is a wave of programmed cell death that helps removing or that is meant to remove those neurons that eventually are not integrated in the network. So practically, the way that this is thought to the meaning of this programmed cell death is that in the brain is made of a higher number of neurons initially than needed. And this to me makes a lot of sense because you don't want to run sort of in limited resources. And then a network is forming, start to be active, and once is sort of at least wired, then those neurons that are not integrating into the network, they are being removed. And this is happening specifically at this time window. And so what we were observing is that in our mutants, we had indeed a higher proportion of programmed cell death compared to the wild type, and then this higher proportion of this window of this increase in apoptosis in the mutants is really limited to this period between P0 to P10, and in fact, within a few days, they have this increase or higher amount of programmed cell death compared to the wild types. So, so practically, what we observe is that in the mutant animals, we have an exaggerated programmed cell death wave, and this is not so. This window and this programmed cell death stage is not just happening in mice, but in fact is also occurring in humans. When in humans, this is happening with a slightly different time frame, as everything in human brain development, it takes much longer. And it has been described to occur between post-conceptional week, approximately 26 weeks after post-conception to up to 6 to 8 months after birth. And so what we did then went back to, in fact, to some additional patients with mutation in SS75, where we could track the development of their head size. And in fact, what we found is that those patients with mutation in SS75, they are born with a sort of slightly below the norm head size. But within the very first 6 to 8 months of age, the head size sort of diverge way more from the average developmental curve. So practically what it means is that those individuals were born with roughly -2 standard deviation of the head size of 2 standard deviation below the norm, and then within 6 to 8 months, the head size gets to a -5 standard deviation. So really indicating that also in humans, it seems that deletion of SS75 leads to this decrease in head size, so this microcephaly in within the time period of this programmed cell death. And so what we next wanted to really understand was whether SS75 is increasing the programmed cell death in a cell-autonomous or non-autonomous fashion. So it's basically, is this a network property, or is the SS75 deletion itself sufficient to lead to this increased, exaggerated programmed cell death? And so that for this, we went downstairs to one of our colleagues, Simone Primaia, that has a very nice system to really quantify very precisely cell-autonomous versus non-cell-autonomous effects. And so practically what you need to know for that is that simply that you are analyzing side by side 2 different mouse models. One is the wild type, where you have randomly some cell coloring green and some cell coloring red. And because this is sort of a random process, because all the cells are wild type, if you make the ratio between green to red cells, you expect that this ratio should be 1, because there is no difference practically between the two cells. And then you have another mouse model where you have instead all cells in the background are heterozygous for their gene of interest, in this case SS75, and then a few cells are red and wild type, and other cells, also few of them, are green and mutant. Now, if the mutation itself has an effect in a cell autonomous manner, if you're going to do on cell either cell proliferation or survival, if you're going to make the ratio between the green to red cells, you may find a different ratio than 1. And so this is exactly what we did with SLC75, and we did quantify this ratio between green and red cells in wild type and mosaic animals at different developmental stages. And what we saw is that at P0, we found really no significant difference between the ratio of green to red cells between the mosaic and the wild type. But then as we move to the P5, indeed, mosaic animals have a lower ratio than 1, indicating lower amount of green cells compared to red cells, indicating that, in fact, mutation of SLC75 leads to probably, as we have shown, in fact, to the removal of some of the neurons in those animals. So really indicating that SLC75 practically is cell autonomous, regulating the capacity of that cell to survive. So when SLC75 is missing, even in one cell, the probability of that cell to survive is lower. And so then, and what we also have shown is that this is quite specific. It's not just specific in terms of time. So as I have shown to you, this is very restricted to the period in mouse between P0 and P10, but it's also restricted in terms of cell types that are affected. So excitatory neurons are affected, and those are the cells where we are deleting SL75. But also interneurons are indirectly affected. Indeed, other people have shown previously that during this time period of cortical network refinement, the interneuron number is adapting based on the excitatory neuron number, so that the ratio between the excitatory neurons and inhibitory neurons always remains stable. And so what we found is in this animal is, despite here we are not deleting SL75 from the interneurons, the interneuron cell number is also decreasing. On the contrary, other cell types, so non-neuronal cell types, including astrocytes and microglia cells, they are not affected in these mutants. And again here, so the nice thing is that interneurons are affecting despite SL75 is not deleted from interneurons, while astrocytes that in principle have also the deletion of SL75 are not affected. So in simple words, this indicates that not only SL75 is cell autonomously affecting the survival of excitatory neurons, but this effect, this function is specific to neurons or specific to excitatory neurons, because in fact it's not affecting glial cells. Next, we ask really how, in fact, does SL75 affect the neuronal survival at early postnatal stages? So we have found that SL75 deletion itself is sufficient to trigger, sort of, if you want, the increase cell apoptosis or programmed cell death of those neuronal cells within this time window. But how is this happening? And so we thought that eventually one way to really explain this is that neural activity may be affected in those mutant animals, and the reason, or mutant cells. And the reason is that, in fact, there were previous work from others that have shown that neural activity may be one means by which the network is measuring whether a cell is sort of included and wired within the network or not, sorry, whether a cell is wired within the network or not. This makes a lot of sense, right? It's sort of a measure of, say, whether that hub is needed or is not needed. And of course, because we knew that lipids were affected, and we know that lipids can affect, as I mentioned before, membrane proteins, as membrane properties, as well as protein function, we thought that eventually neuronal activity could be a good way to explain why SS75 mutant cells may be affected. And so we proceeded to test that by using patch clamp analysis and measuring really just the neuronal excitability of those mutant neurons. And in fact, so we did this experiment also in the cortex at P6 to P7, so measuring the number of spikes that we were getting in the wild type and SS75 mutant cells or mice on the same amount of injected current. And as you can see in the curve on the left graph, is that for the same amount of injected current, the SLC75 mutant cells, they are spiking less than the wild type cells. And this despite the fact that, in fact, the shape and the quality of the action potential that we observe in those mutant cells is pretty much is not of indicating of just of a neuron that is immature neuron, but rather say that those neurons can spike eventually with the proper shape or proper in a proper way, but they are firing those cells are less excitable. Yet here we still had one problem, and namely was the fact that we knew that already at this time point in the mutant animals we did have fewer neurons than in wild type animals. So we thought that eventually the effect that we were seeing could be explained also by sort of intrinsic plasticity defects. Although we would not have expected to go in this direction exactly, because you expect just to be compensatory, so eventually to go in the other direction, still this was a possibility. And so the way we checked this is we went back to this mosaic system model where we checked before this MADAM system, where we checked the cell autonomous effect on neuronal cell survival of SLC75. And in this case, we could patch side by side wild type in mutant cells in a heterozygous background. So really meaning that the network is the same. And so now we were measuring the excitability of those cells and really linking the excitability of the cell with the mutation of SLC75 in that specific cell. And in fact, what we found is, as you can see, that green cells still are firing less than red cells. So really indicating that mutating SLC75 cell autonomously control or determines the frequency or the excitability of those cells. Finally, also because I mentioned that all the effects that we observed in the network refinement as well as in the lipid composition was an effect limited to a specific time point, so namely between P0 and P10, we wanted to check whether this excitability phenotype was something that would stay longer or it was also confined to this time period. And so we patched then cells at the later stages, and this somehow it's not appearing here, but what I can tell you is indeed when we are checking at the later stages, the firing rate of those neurons lacking SLC75 was comparable to wild type. So similarly to the lipid effects, to the programmed cell death, and all the analysis that we have done, also the excitability of the neurons is affected really just at that very specific time point. Nevertheless, those animals have sort of sustained behavioral defects. And this is, and I'm not going to go through all of this because it's really just to give you an idea, the fact that even if this effect of SLC75 is really confined to a specific time period, then of course they can have a sort of sustained defect. So with this, I'm going to summarize and summarize what I told you. And practically what I've shown to you is that the metabolic program of the brain is obviously very complex. and that just measuring the level of those metabolites, we can define different metabolic states of neuronal cells and non-neuronal cells as well of the cerebral cortex across time. Obviously, also here something's missing. What I was showing to you is that eventually a defects in the so that the branching amino acids actually they are most probably needed to support the production of energy of the neurons at a very specific stage when glial cells are not present yet in the brain or not yet ready. They are present, they are actually there, but they are not yet eventually completely supporting the neurons. And so at that stage, the neurons is utilizing additional source of energies, namely the branching amino acids. When we are deleting the branching amino acid transporter SS75, what we found is that the neurons start utilizing the lipids to provide energy. And probably, and this is something that, in fact, is very interesting, and they haven't, it wasn't one picture before that you couldn't see, but is very interesting is that the branching amino acid catabolism and the lipid catabolism are sharing a number of different enzymes. So really practically telling us eventually that those two pathways are somehow linked, they are physically sort of linked to each other. And then this probably is really to try to make compensatory mechanism for the neurons to properly function. And in the presence of mutation of SS75, the drop of branching amino acid, drop of lipids, we then identify these changes in excitability of the cell that we think they're most probably due to post-translational modification of proteins such as ion channels that can be regulated by, for instance, lipid modifications such as palmitoylation, and that this may be leading to the defects in excitability that we observed. I hope that at least, one other thing that I really wanted to comment on is really that we think that eventually the metabolism may be really a potential nice link between the environment and so extrinsic and intrinsic properties of the brain. And so that maybe is one really way of moving or changing the penetrance of different mutations. And so that's what we are working on. And with that, I really hope that at least this slide is, it looks quite okay, because this is the most important. This is really mostly the work of a former PhD student in the lab, Lisa Knaus, that did an excellent work. As you have seen, this was primarily her work with the help of some other people, such as Bernadette in the lab, Christoph, and a few others, but you see most of them here. And of course, we also had some collaboration. I mentioned Simon, but there is a long list of people that really helped us with this project. So with that, I hope that was a bit unfortunate about this messy slides, but I hope that at least you could follow a little bit the story. So thank you.
Thank you, Gaia. It was a very fantastic presentation. I really enjoyed it. Okay, so now we move on to the questions. We have a question in the chat from by Sulov Saha. They say, great work. Did an increase in acylglycerol manage to compensate any brain defects?
Well, so practically we don't know. So thank you. This is a good question. Practically we don't know in the sense that I believe so, but the only way to know would be to now knock out, for instance, or knock down or inhibit somehow those proteins that seems that may be the connection between the brain and the rest of the body, such as GCN2, or to the proteins that eventually they are helping the triglycerols to travel into the brain. So my assumption is that if we would do so, indeed we would have much worse phenotypes, but it's just an assumption. I cannot, so a speculation. I cannot, we didn't do this experiment.
I see. Then we have a couple of questions by Sylvie Henrique. She says, nice talk, Gaia. You mentioned that the increase in triglycerides in the brain can be due to the brain to sense lack of energy and cause the liver, for example, to ramp up lipid synthesis. However, given that you are mutating an amino acid transporter, I would expect that similarly that would cause an increase in amino acid appetite, given that the brain has less access to branched-chain amino acids. Did you test that or observed increased appetite for protein or food in general in these mutants? Thank you.
Yeah, so that's also a very good question, and so I should mention a couple of things. So the first thing is that so we are already humans at least, but also mice eventually. We are already getting way more amino acids, at least branching amino acids, that we need through our diet. So I think the calculation last time I made it was something like 10 times more amino acids at least than what we need. So now everything then runs on the properties of SL75. So it can be explained by the property of SL75. So the Km value of SL75 is very low, meaning that at a normal plasma concentration of branching amino acids, the transporter is completely saturated. And in fact, inside the brain, the level of amino acids is about 10 times less of what is in our plasma. So now, if I would just increase the level of plasma amino acids, I would practically not change anything, because SL75 is still at the barrier, is still limiting the transport of branching amino acids and keeping them in place. And if I would even pass by the barrier and put them through, which we have done in the past, do intraventricular injections of those amino acids, those amino acids would still not be able to go through the neurons. So in practice, I know this is just, I know that at least in humans, there seems to be some report that in fact this is also increasing protein requirement when in the presence of those mutations. But eventually, if you have transporters, mutation in the transporters, this increase in uptake, sort of dietary uptake, will be insufficient to provide more amino acids to the brain. So it was a long answer to say yes. Eventually, there are also those mechanisms, but they do not work eventually, because then the main limiting step is really the transporter itself.
I see. I had a similar question. I was wondering if there were other transporters for branched-chain amino acids that could somehow compensate if you provide the nutrients in the diet.
There are other SLCs, and in fact, I haven't been completely, because there are so many amino acids, I tended to simplify things, and there are other transporters, and in fact, the SL75 does not transport only branching amino acids, but also some other amino acids, and in fact, it stands for large neutral amino acid because it transports also those. And the point is that based on a number of the analysis that we have done, and practically based on different knockout mutants that we have done, what we find is that the only one that are really very impaired in those mutants are the branching amino acids and beta-methionine. And so what we think is that really that while there is while those transporters are sort of promiscuous in general, it seems that the branching amino acids, they really rely on SL75. And of note, it's also nice to see that that's why I refer to this Km value, is that other transporters have different Km, and usually they have higher value, meaning that eventually they can lead to a more less saturated transport. And so the properties of all those transporters also kind of counts toward amino acids and what amino acids are transported.
I see. We have another question by Sylvia. She says, you believe that after P2, BCAAs are necessary for energy, but could these phenotypes be caused by impaired protein biosynthesis, which may be particularly important to develop the brain after birth?
Totally. So this was I think our first hypothesis, like the mTOR protein synthesis and all of this is really something that we really, and in fact, I felt that in part this was going to be the most boring project ever, because to me it was sort of obvious, and it was surprising to see that this was not the case. So I didn't show it here, but first of all, we checked a number of other so a number of proteins related to protein synthesis, and we did not find any significant change there. We did a proteomic analysis because we really couldn't figure out why was that, and also there we couldn't find any difference. So now my sort of understanding of that, and it's of course it's not the easiest, but something that we need to work on, is that the cell eventually is capable of prioritizing. So here in one of the graphs I would have wanted to show to you, it was that the level of the branching amino acids is reduced, but it's not that there are zero branching amino acids. So there are still there is a considerable amount of those amino acids. And so my understanding, the way I would interpret this is that, again, depending on the level of amino acids, you sort of prioritize the amino acids toward, and this, again, it's probably all biochemical type of properties of all the enzymes that are involved in the different pathways. The cell is sort of prioritizing what functions, where the amino acids should go, right? And so that things that have higher affinities, they will take a certain amount of branching amino acids, either lower or higher amounts. And so that at the end, what seems to happen to me is that obviously those mice and those humans that are alive, they're fine, and they're producing proteins normally. And so that means that all those amino acids probably they're going through that path. And the energy part is that the one that sort of suffered the most, and that's how it's compensatory and so on.
Very interesting. Okay, I think we're done with the questions. Now we have the Q&A session on Zoom for people to get to know Gaia a little bit more and to delve into deeper discussions for like 20 minutes. So yes, I'll see you there. The Zoom link is on the chat box here in the Crowdcast. So yeah, with this, I would like to thank Professor Novarino once again for an amazing talk and a great discussion as well. And I'll see you all soon. Thank you. Bye-bye.
Bye.